Hello everyone, my name is Sumit. I am back with the new video. Today we will discuss about the antigens and the antibodies. First is antigens. Antigens are large molecules of proteins present on the surface of the pathogen, such as bacteria, fungi, viruses and other foreign particles. When these harmful agents enter the body, it induces an immune response in the body for the production of the antibodies. For example, when a common cold virus enters in our body, it causes the body to produce antibodies to prevent from getting sick. Then we will talk about the properties of antigens. The properties of antigens are: antigen should be a foreign substance to induce an immune response. Antigens have a molecular mass of 40,000 to 6 lakhs Dalton. They are mainly proteins and polysaccharides. They are more chemically complex, they are more immunological they will be. Antigens are a specific species. The age influences the immunogenicity. Very young and very old people exhibit very low immunogenicity. Then we will talk about the types of antigens on the basis of origin. There are different types of antigen on the basis of origin. First one is exogenous antigen. Exogenous antigens are the external antigens that enter our body from the outside. Example, inhalation, ingestion, etc. This includes food allergens, pollen, aerosols, etc. and are the most common type of antigen. Endogenous antigens. Endogenous antigens are generated inside the body due to the viral or bacterial infection or a cellular metabolism. Then next is autoantigens. Autoantigens are the self proteins or a nucleic acid that due to some genetic or environmental alteration get attacked by their own immunity system, causing the autoimmune disease. Then next is tumor antigens. It is a antigenic substance present on the surface of tumor cells that induce an immune response in the host. Example, MSC molecule first and MSC molecule second. Many tumors develop a mechanism to evade the immune system of our body. Then next is native antigens. Native antigens, that is not yet processed by an antigen presenting cell, is known as the native antigens. On the basis of immune response on the basis of immune response, antigen can be classified as: first one is immunogen. This may be protein or polysaccharide can generate an immune response on their own. The next is hapten. These are a non-protein foreign substance that require a carrier molecule to induce an immune response. Then next we will talk about the antibodies. Antibodies, AB, is also known as immunoglobulins. These are large Y-shaped blood proteins produced by the plasma cells. They bind to foreign particles and invade them, means to inhibit them. These particles are foreign bodies that get attacked by the antibody. Antigens are foreign pathogens that invade the body and have the capability to give rise to a response from our immune system or immunity system, either by grouping up with the larger molecule or alone after binding with antibodies for a particular immune response. Hence, the antigens stimulate the production of the antibodies by the immune system. An antibody has a Y-shaped structure made up of 4 polypeptide subunits. Each subunit has two identical light and heavy chains. The N-terminal of each heavy chains forms an antigen binding a domain with a light chain. There are two antigen binding domain forming the arms of the Y shape. They are known as fragment antigen binding, that is FAP domains. The C terminus of the heavy chains from a fragment crystallization, that is FC domain, which helps in the interaction with the effector cells. All four polypeptide subunits are held together by disulfide and non-covalent bonds. They have heavy chains of the antibodies contain a variable region and three constant regions. Each antibody has two identical antigen binding sites and they differ in the antibodies. Then we will talk about the types of antibodies. Antibodies or immunoglobulins are five different isotopes. This classification on the basis of H or hydrogen chains. Let's look at the different types of immunoglobulins and their function. In that, first one is IgM. IgM is the first antibody produced in a response to a microbial attack by a beta cells. It is the largest antibody and it is found in a pentameric form. It circulates in the blood and lymph and constitutes 6% of a total antibody contained in the serum. The next antibody is IgG, most abundant isotype in the plasma and comprises 80% of the total antibody contained in the serum. It detoxifies substances that are harmful and recognize the antibodies and antigen complexes. It is transferred to the placenta through the fetus and protects the infant until its birth. The next antibody is IgA, usually found in a liquid such as breast milk, serum, saliva, fluids of the intestine. IgA in a breast milk protects an infant gastrointestinal tract from the microbial activity. It consists 30% of the total antibody contained in the serum and it is divided into two subclasses, IgA1 and IgA2. Among these, IgA1 is highly found in the secretion and is also called as the secretory immunoglobulins. The next antibody is IgD. It is involved in the production of the antibodies by the beta cells. It is present as a monomer and weigh around 1,80,000 Dalton. It comprises less than 1% of the total antibody contained in a serum. Then next antibody is IgE. It is present in the least amounts, around 0.02% of the antibody contained in the serum. These are present in the linings of the respiratory and the intestinal tract and it responds to the allergic reaction. Then we will talk about the function of the antibodies. First function is bind to the pathogen. The next function is activities or activates the immune system in case of bacterial pathogen, directly attacks viral pathogens, assists in the phagocytosis. Antibodies provides long-term protection against the pathogen because it persists for years after the presence of the antigen. It neutralizes the bacterial toxins and binds to the antigen to enhance its efficacy. Then they also act as the first line of defense for a mucosal surface. They ingest cell by the phagocytosis. Few antibodies bind the antigen present on the pathogens. This aggregates the pathogen and they remain in our secretions. When secretion is expelled out, the antigen is also expelled. This is all about the antigen, its function, types, antibodies, function and its types. Thank you.